So let's talk about protein basics. This video is aimed for people that don't quite know what the various loops and coily things are. So hopefully this video will make things clearer. So firstly, a protein is a 3D structure composed of one or more peptide chains. A peptide is a series of amino acids linked together with a peptide bond, as shown over here. You've got nitrogen that's bound to a carboxylate. So, and an amino acid in a chain is called a residue and can have either and it's got two parts, the backbone, which forms this chain, and the side chain, which is unique to each amino acid type. Now about those wiggles. So the spirally things are alpha helices, and that's because the backbone is forming a hydrogen bond to the backbone of a residue that's free ahead or free behind of it. And the flat strips things, over here we've got three in anti-parallel formation. These are beta sheets. The backbones are forming hydrogen bonds to another sheet. And then we've got the bits that are neither alpha helices or beta sheets, these loopy things, which loops or disordered regions. Although not all loopy things are disordered, as some of them are quite tight turns, are very rigid. Additionally, we've got bits that are not part of the protein, which are ligands. These can be small molecules which are covalently bound or non-covalently bound, and are represented using normal chemistry colors, say blue for nitrogens, red for oxygens, and carbons are whatever color you fancy. Sulfurs are colored in yellow, whereas phosphates appear in orange. So we've currently seen two different types of representation. One is the cartoon mode, this very streamlined mode where the helices and sheets are quite exaggerated. And we've also got this atomistic, where you can see each of the atoms. And one thing that is worth pointing out is that in crystal structures, sometimes you've got waters which are coordinated in such a way that they actually are part of the crystal. They're solid, they don't move. And as a result, you will see waters in your protein crystal. And as you can see over here, there's lots of oxygens. As with X-ray crystallography, electron crystallography, we don't have electron being converted to X-ray, sorry. You don't actually see protons, and as a result, all the water molecules, we can't see the protons. Although, if you're lucky and have a neutron model, you will see the protons. And obviously, in molecular simulations, in computed models, you can obviously see the hydrogens. So hopefully this all made things clearer, and now you know what those wiggles are. Thank you.